بقول হ্যালো, করি নাই কিন্তু
আমার
কোনো
আমি
আওয়াজ
স্যার
আসছে না,
সেকেন্ড
শুরু
কম্পিউটার
করি নাই।
নরমাল থেকে চালানি কি মধ্যে আইছে
স্যার আসবে স্যার একটু টেকনিক্যাল
আচ্ছা, ঠিক
প্রবলেম
আছে।
হচ্ছে স্যার আসবে হয়ে যাবে স্যার 1-2 মিনিটে হয়ে যাবে হ্যাঁ
তাই জন্য একবার আরম্ভ হলে
Uh, Very good morning. We are going live now. Sorry that there was a delay for some network glitch from this end. So we can initiate the process. We are going live in few seconds now from now. The hosts are to take
কেমন আছেন?
offer to the host.
ভাইস চ্যান্সেলর স্যার রেডি হয়েছে স্যার আমরা রেডি এখন উই আর রেডি স্যার হ্যালো দেবাশিস দেবাশিস দেবাশিস
Tulal, please go start.
দেবাশিস
Yes, yes,
ক্যান ইউ হিয়ার মি দেবাশিস
yes, yes. Hear you.
ওকে
VC স্যারেরটা শোনা যাচ্ছে না।
স্যার ভিসি স্যার শোনা যাচ্ছে স্যার হ্যালো হ্যালো না হ্যালো ভিসি স্যার শোনা যাচ্ছে স্যার হ্যালো
I think there is a problem with the লাগবে না কিছু করতে হবে
সুদীপ 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 হ্যালো সুদীপ 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 হ্যালো সুদীপ
Yes, I can hear all. I can hear all. All the sounds are clear and loud.
ভিসি স্যার শুনতে পাচ্ছেন স্যার ভিসি স্যার শুনতে পাচ্ছেন স্যার
I think his microphone is on mute mode. The microphone has been muted.
স্যার আমি যে কথা
সাউন্ড আসছে না প্রফেসর মন্ডল।
হ্যাঁ আসছে এখন ঠিক আছে hear you. I can hear you sir.
সাউন্ড আসছে একদম ডিম সাউন্ড আসছে, অডিও প্রবলেম।
হ্যাঁ স্যার হ্যালো স্যার এখন শুনতে পাচ্ছেন স্যার হ্যালো হ্যালো যে কথাটা বলি এটা 1-1.5 মিনিট পরে
ইন্টারনেটে লেগে যায় কিছু করার নাই না এটা আপনার না স্যার কিছু এটা আপনি যেটা করবা লাইভে একটু করে কিছু করার নাই স্যার
অনেক অনেক
কতজন শেষ করবো
only 17 only 17 মনে ওগুলোর জন্য হচ্ছে ইউটিউব গুলো বন্ধ করে দিতে
আমরা লিখে আছি এটার মধ্যে একটা কানেক্ট ফোন করতাম
আমি কথা বললে 1 মিনিট পরে আসে এক মিনিট পরে আসে
এক মিনিট 1 মিনিট পরে আসছে আপনারা তো ঠিক আছে
Are we audible now?
আচ্ছা স্যার শুরু করতে
Are we audible now?
yes
Yes.
Hear me? Can everyone hear me?
Very loud and clear. Very clear.
Okay.
The audio is loud and clear.
Now it's okay.
Devashish
Yes, we are starting.
Sudeep, audio is clear? Sudeep?
Clear audio.
Okay.
Clear.
আমি দুইবার শুনছি তো আমি প্রবলেমটা কথা বলতে অসুবিধা হবে তোমার
যারা মাইক্রোফোন সবার অন্ত স্যার 17 জন মাইক্রোফোন অন্ত এটাই কি বললে আমি দুটারে কইয়া তাই স্যার ইকোর অফিস ওই তো বাকিরা সব করে দিতে বেটার স্যার
Abhijit, you have to switch off you have to mute your thing. Abhijit.
তোর কথা আসছে
Good morning, ladies and gentlemen. I, Devdulal Sharma, on behalf of the Department of Chemistry, Assam University, Silchar, extend a hearty and warm welcome to one and all attending this international webinar organized by the department as a commemoration of its Silver Jubilee celebration. The topic of the webinar, Antiviral Drug Discovery, Creation of an Armory for the Present and Future Pandemics, is very relevant as far as current scenario is concerned. Humankind has seen many a crisis since historical times and has also survived those through collective effort and perseverance. The year 2020 has placed before humanity another such crisis in the form of the novel coronavirus. As mankind continues to struggle to contain the spread of the virus and look for a possible cure, several pertinent questions have cropped forth, which needs the attention of all and sundry. The current pandemic has found the world unprepared by all means, whether it is in terms of effective healthcare or constructive scientific research. The question is how we, as people of science, can contribute to alleviate the current crisis and suggest possible remedial measures for future pandemics. With advancement in science and technology and associated computational techniques, antiviral drug discovery has become a much more versatile process.
Remedial measures for future. With advancement in science and technology associated computational techniques, antiviral drug discovery has become a much more versatile process. With a combination in silico, in vitro, and in vivo techniques, it is possible now to screen a large library of compounds, natural as well as synthetic, for their antiviral activities in much shorter duration. However, much more scientific artistry is involved in the entire process, which the current webinar is expected to address. I would like to introduce our audience to our guest speakers for today's event. We have with us Dr. Aviva Levina from the University of Sydney, Australia, Professor Ritu Gaur from South Asian University, New Delhi, India, Dr. Giacomo Rena from CNRS Delegation Alsace, Strasbourg, Alsace, France, and Professor Ayushman Sen from Pennsylvania State University, USA. We are extremely honored by your presence. I would now like to request the head of the Department of Chemistry, Assam University, and also chairman of today's webinar, Professor Paritosh Mandal, to share a few lines. Sir, please.
Very good morning to all of you. Honorable Vice Chancellor, Professor Dilip Chandra Nath, our guest of honor, Professor Somnath Dasgupta from IJAR Kolkata, and ex-Vice Chancellor of our university. Professor Somnath Dasgupta has recently received the prestigious Asiatic Society Lecturership Award 2020. Congratulations you, sir. Professor C.R. Bhattacharya, Dean, Albert Einstein School of Physical Sciences, Dr. Devashish Sangupta, Dr. Sudeep Chaudhary, convener of this international webinar. International speaker, Professor Ayushman Sen from Pennsylvania State University, USA, Dr. Aviva Levina from University of Sydney, Australia, Dr. Giacomo Rena, CNRS Delegation, Strasbourg, France, Professor Ritu Gaur, South Asian University, New Delhi, honorable distinguished guest, participant from different colleges, universities, and institute of this country and abroad. Colleague from Assam University fraternity, research scholar, students, ladies and gentlemen. I take this privilege to welcome you all to the international webinar on the topic Antiviral Drug Discovery, Creation of an Armory for the Present and Future Pandemics, organized by the Department of Chemistry, Assam University, Silchar. It gives me immense pride to mention that the Department of Chemistry, Assam University, Silchar, India has entered the 25th year, that is the Silver Jubilee year in the year 2020. As part of the Silver Jubilee celebration of the department, we are organizing this international webinar on the pertinent topic of the that can probably make a meaningful attempt to motivate scientific research by strengthening therapeutics and immunogenic responses to viral and microbial attacks. Mankind's survival on this planet rests at the hand of our scientific community. Therefore, we need to popularize competent scientists to ensure the safety and security of our individual, the society, the nation, and the globe in general. While every citizen will now have a role to play an important awareness, science will have to take a front seat to prepare mankind for greater challenges. Nations and their leaders will have to take cognizance of this fact that invest upon scientific research more magnanimously on a priority basis. From the beginning of 2020, the COVID-19 pandemic has created a massive crisis not only in India but all over the world by paralyzing social, economic, and academic life. We are confident of our abilities to overcome this crisis as civilization has overcome pandemics of the past, like the Spanish flu and many others over the times. Recently, an outbreak of another novel Bunia virus, which is spread by ticks, has been identified once again in China. So, our resilience during these challenging times will be tested on the basis how we combat the spread of virus and how we can make the best out of the circumstances. The purpose of this webinar is to create a small platform to showcase the relevance of developing therapeutics to cope with the need for antiviral drugs. In this regard, chemistry has occupied the middle ground between physics and biology, and it deals with the organization and reorganization of atoms into the myriad of combinations that distinguishes one molecule from the next, rather than the fundamental nature of matter or its animation in time and space. Modern perspective of material research is the prolific attribution of fundamental concept of science inherited through ages to miniature technological design. On the other hand, chemical research in biological and medicinal sciences deals with the development of pathogenic detection, biomedicine, biological sensing, electronics, imaging, biochemical and biophysical properties of DNA and RNA, photodynamic therapy of cancerous cell, etc. Understanding the chemistry under earth, these biological processes is indispensable to bring out newer perspective for further research. Recent advances in the synthetic methodologies, characterization, computational and simulation techniques, and diverse application in material and biological sciences have created a newer paradigm towards the development of indigenous materials and methods for the service of mankind. Most of the discoveries in the history of science has been augmented from the critical retrospection of the most simple and general idea and experimental finding that lead to the complementary to each other. Deliberation in this webinar will provide an opportunity to share the latest exciting discoveries and inventions and design a landscape for contemporary chemists towards the newer dimension and horizon of research that merges material and biological sciences from both theoretical and experimental perspective. Pioneer or leaders are those who can think ahead of time. We are very fortunate that the present webinar has been illuminated by the auspicious presence of internationally renowned scientists and researchers, those who are pioneers in the present scientific parlance to elucidate the role of chemistry in diverse arenas of material, biological, and medicinal sciences. As William Bragg said, I quote, "The important thing in science is not so much to obtain new facts as to discover new ways of thinking about them. The webinar deliberation will provide an initiative, intuitive, efficient, and useful platform, especially for young researchers to enhance their wisdom and creativity, knowledge, sportsmanship towards the pursuance of science as their career and for portraying science in the realm of life. The Department of Chemistry, established in the year 1996, is vibrant in imparting teaching through an advanced academic curriculum, as well as conducting researches in frontier areas of chemical sciences, reflected through quite a good number of publications in each year in reputed national and international high-impact journals. A friendly relation existing among the faculty and staff member, research scholar, students is a natural wealth of this department. In recent years, our faculty members have received several projects from different funding agencies like DST, CSIR, UGC, DBT, BRNS, DAE, DRDO, etc. The Department of Chemistry also received UGC SAFE, DRS, and FIST DST, plus a Center for Soap Matter granted by UGC 11 plan, bears testimony to our humble growth. I take this opportunity to congratulate all ex-colleagues and alumni of the department for their contribution to the growth and development of the department. I am extremely honored to welcome all the distinguished guests, invited speaker, and other participants to this international webinar. I am hopeful that all the effort to organize this event are going to be a grand success. Thank you very much.
Now I would like to request the Dean, School of Physical Sciences, Assam University, and patron of this webinar, Professor Chiroranjan Bhattacharjee, to share his thoughts with us. Sir, please.
Thank you. At the very outset, seize this opportunity to express immense pleasure to have amongst us Professor Somnath Dasgupta, Senior Scientist, ISAR, Kolkata, as guest of honor for this international webinar, who has also been the Vice Chancellor of our university in the recent past. For being conferred upon the prestigious Asiatic Society Lectureship Award 2020. Congratulations, sir. Honorable Vice Chancellor, Professor Dilip Chandranath, Professor Paritosh Mondal, Head of the Department, Professor Pradeep Chandrapal, Organizing Secretary, Dr. Devashish Sengupta, Dr. Sudeep Chaudhary, the conveners of the webinar, esteemed invited speakers of this international webinar, Dr. Aviva Levina, the University of Sydney, Australia, Professor Ritu Gaur, South Asian University, India, Dr. Giacomo Reina, Institute of Cellular and Molecular Biology, CNRS, France, Professor Ayushman Shen, the Pennsylvania State University, USA, colleagues, staff members, scholars, and dear students. In the department, colleagues from allied departments in the university, dear participants from various academic institutes and universities, I extend warm welcome to this international webinar on antiviral drug discovery, creation of armory for the present and future pandemics, being conducted by the Department of Chemistry under the Albert Einstein School of Physical Science. Established way back in 1996 in a geographically remote corner of the country, this year the department has stepped into the 25th year of its existence. The first batch of students were admitted into the PG program of the department beginning July 1996. Given the very critical time we are passing through, owing to the global pandemic caused by the coronavirus, the confluence of this international webinar on antivirals and the celebration of Silver Jubilee has created a splendid ambience. Students and scholars passing out from its portal over the years have found respectable space across the nation and world, contributing in diverse domains. I congratulate the entire department, faculties, scholars, students, and staff who are witness to this historic milestone today. I also recall the contribution of the founding faculty members, distinguished visiting teachers, all the alumni, and many others for their immense contribution in developing, taking this department to a new height. Over the course of human civilization, viral infections have caused millions of human casualties worldwide, driving the development of antiviral drugs. A new era of antiviral drug development actually began with the antiviral drug hydroxyridine in June 1963. In a span of little over 5 decades, more than 90 antiviral drugs got approved to treat 9 types of human infections caused by DNA, RNA, or retrovirus. While several antivirals have been developed to fight against current and emerging infectious diseases, as of April 2016, many antiviral drugs and or vaccines have been approved for the treatment of 22 infectious diseases: HIV, HBV, HCV, HCMV, HPV, HSV, RSV, VZV, influenza virus, smallpox, rotavirus, human adenovirus, human herpes virus 6, polio virus, hepatitis A virus, hepatitis E virus, Japanese encephalitis virus, yellow fever, rubella, measles, mumps, and rabies. Nevertheless, there is still no antiviral drug or vaccine for more than 200 infectious diseases that are afflicting the human population worldwide currently. Despite the rapid advancement of pharmaceuticals and biotechnological approaches, the development of successful antiviral for safe treatments remains a challenge. Potent antiviral drugs that counteract the highly variable nature of virus genomes are still required because emerging drug resistance, narrow therapeutic index remain a major cause of treatment failure. Completely remove viral components integrated into the host cells as well as kill the virus itself are required in future antiviral development. The title of this international webinar, which mentions about creation of armory of antiviral, is in itself very significant theme. I commend the organizers for choosing such an apt, timely theme for the webinar and their efforts to get eminent researchers to deliberate on the latest development in the field. Fusion of experimental with efforts has remarkably advanced the pursuit of newer antivirals. Search for newer drug molecules, vaccines, repurposing existing drugs vis-a-vis the societal political dynamics world over are likely to pose severe challenges for the entire mankind in the forthcoming years. No great thing come out of the blue. With the sustained effort from the continuing inflow of talented young scientists and the growing effectiveness of the research community, look forward to more and more pioneering achievements in the domain of antivirals. Thus, I candidly believe that this webinar being organized in this critical time, severely affected by the global pandemic, unseen ever before, will inspire the new generation researchers profusely. There is also an urgent need for forging meaningful collaboration of scientists working in diverse disciplines and domains to address these important issues. Wishing the webinar a great success, I once again heartily welcome distinguished speakers, all the participants, and our guest of honor, including our Honorable Vice Chancellor. Thank you all.
I would like to call upon our Honorable Vice Chancellor of Assam University and also the Chief Patron of today's webinar, Professor Dilip Chandra Nath, to share his views. Sir, please.
হ্যালো, কোনো সাউন্ডই আসছে না, অডিও ইজ মিসিং। ওকে ওকে, দ্যাটস ফাইন। Hello, Namaskar. Good morning to everybody. Actually, I am facing problem. Many times I am not hearing anybody's. So this is a problem. Anyway, today is a very special day for us. My PC reader, Professor Somnath Dasgupta, with us, and the Assam University congratulate him. You know, the reason, you know, very obvious. He got the award, Asiatic Society Award. This is a very important and I history, specifically the Professor Mandal and Sudip Chaudhuri, Devashish Sengupta, and Dean C.R. Bhattacharjee, who took pain for organizing this and you know, gathering many you know, resourceful persons deliver their talks, and more especially Professor Somnath Sengupta was here this morning. You are most actually would be happy to see you, you know, physically, but the time is like that. We know this is interaction, and COVID is threatening us. Last 1 century, from 1918 to today, we have so many the virus and affecting our health, affecting our economics, affecting our social life. Spanish flu, Asian flu, then SARS, swine flu, Ebola, MERS, and COVID. Now, why this virus and we are affected? So this we have to find solution to prevention of this the virus to come to the society and affecting our health. And there is the kind of medicine or the vaccine, or can you think of how can stop these things? What are the difficulties with the human society? Which region it is coming, and whether future any other pandemic will be there in front of us. One thing may be that we are degrading, our environment. We are making land for us, cutting trees, destroying rivers, destroying hills. So animals are coming close to the human body. Maybe the quick transition with the human. We have to think about that. Preserve this, sustainable situation that animal can live there of their own, and human can live. Also, if the virus comes to the animal, can we treat them? Can we in advance? So there is such transition. Also, our behavior of human people, nowadays throughout the world, the Indian people, anything and everything, any types of meats are available. The killing animals that sometimes without whatever may raw meat. So these are the, when we see that these are to the human population. So we the globally even how the advanced warning and our scientists are here. This is work for the future and the present. Present we are vaccine is first, maybe within a month or so it will available. I don't know, the paper times is a not be correct one, but this is not a solution. Definitely future is there. The chemistry people with biological many scientists to get together and find out what to do for the future. And we have a new education policy now. We are going to implement it soon. The hiccups are there. We are and our teachers also need to be trained. Who classroom may not be good in the virtual classroom. So we need a proper observation and training for them that friends get benefited, the systems get benefited, they can learn. The one problem is our laboratory, the wet laboratory lab basically. So the government is thinking that the virtual lab can be developed, and that will be done at some stages, but everything cannot be the virtual, is a kind of mixing. But initial stage when we cannot go to the lab, when we cannot, perform experiment in the laboratory, then we have to think initially the virtual. But also we should be, active. It is not that that we cannot go to the lab. We have to go by some means, maintaining some SOP. When the police people can work, the health workers can work, the Safai Karmi can work. We also have to follow a kind of SOP, and we have to. I am in favor of opening the laboratory following the SOP. Once the government clearance come, we can think of the opening our lab. Some kind of partially, virtually, and partial in the physical sense. Because if we cannot work, how can we find a solution? So we need to work hard. It is not the fearing and the psychic thing that if we go there, it will affect me when other people can go. But we have to keep our health okay, and we have to maintain the the guidelines of WHO and maintaining all this. Possibly we can progress our education, but it needs a kind of training and SOP, whatever the health people tell, and we follow that. But we cannot stop. We cannot hold inside the your own home or residence, and the only section of people will work. So we have to work in some way and progress our education and progress our economy. So we have some time limitation. You understand? The beginning I was understanding joining, but anyway, I will be here for another 15-20 minutes. I will hear Professor Dasgupta, and then possibly I will switch off to another webinar of the university, because we have started the webinar, and every day the Assam University has some kind of webinar going. Today is the 2th, chemistry and the library science, they have another webinar. So tomorrow another webinar. It's a good thing that we are doing something, for the cause of the education. Thank you. Let it be a very successful webinar and good discussion. I understand the students should basically come forward and interact. The research scholars should come forward. They interact with the research persons. I understand there are many good research persons like Dr. Abiba, Professor Ritu Gaur, Professor Reina, and Professor Shen, and others are also with us. With this, I think this our the seminar get inaugurated, and let us progress this seminar at the beginning, and we can have a more intensive seminar, maybe 2 days or 3 days, and we can do it to find the solution and progress our education. Thank you all. Namaskar. Jai Hind. Professor Mandal, I am done. Hand over.
Thank you, sir.
We are privileged to have among us former Vice Chancellor, Assam University, and Fellow of Indian National Science Academy, and currently a Senior Scientist at Indian Institute of Science Education and Research, IISER, Kolkata, Professor Somnath Dasgupta, as guest of honor for today's event. I would request Professor Dasgupta to address this gathering. Sir, please.
Thank you very much. Am I audible to everybody?
Yes, sir.
Yes, sir, you're audible.
Okay. Thank you very much, distinguished scientists from all over the world. Professor Dilip Chandranath, Honorable Vice Chancellor of Assam University, Professor C.R. Bhattacharjee, Dean, Professor Paritosh Mandal, Head of the Department of Chemistry, and my colleagues from Assam University. It's a great pleasure to meet you here in this webinar, although this is a virtual meet. I do hope that, as Professor Nath was saying, that sometime in future we can meet physically and discuss things. In any case, I am very grateful for the invitation and for your kind words that all of you mentioned previous to me. Thank you very much. I am delighted to be a part of this webinar, which is perhaps addressing the most relevant topic of the present time. I am really thankful to the organizers for remembering me and for inviting me to this webinar. I am a non-expert, so to say, and I can hardly contribute in the scientific developments in this particular context. But this pandemic is not just an issue for chemists, physicists, biologists, or the medical community. It's a social issue as well. And it has far-reaching effects on the social structure itself, not only of our country, but all over the world. as a result, it concerns everybody. And so this also justifies my participation in the webinar. Not, even if it is not that much scientific, but as a social animal, as a part of the global human community, I am concerned. Now, I'm very happy to note that this webinar actually coincides to commemorate Silver's Jubilee of the establishment of the Department of Chemistry at Assam University. I have truly great admiration for this department, its faculty members, its research scholars, and its students. And this I say from my personal experience. COVID-19 took us by surprise, and it became very clear that there are a lot of confusions regarding mode of transmission, symptoms, and recovery process. There were lots of conflicting claims that confused us. All of us know that even the WHO changed their stance quite commonly. Of course, this happens in every aspect of science, but in this case of pandemic, this really confused us to a very large extent. This resulted in social discrimination. And in India, we have observed that people being harassed by fellow citizens because of very unfortunate things that happened. Like everywhere in the world, we were also having inadequate healthcare facilities, particularly to tackle this virus. All these issues highlight the importance of this webinar that we are having today, which is aimed at developing an integrated research platform involving chemists, biologists, physicists, and data scientists, and to address issues like therapeutic, diagnostic, and vaccine development related problems with respect to COVID-19. I understand that finance is not a problem all over the world for doing research on vaccine development. All the governments of the world are putting in a lot of money into their efforts. So it is left to us to launch an integrated mission. I hope that at the end of the day, we will arrive at a definite strategy. I am certain that the vaccine will be available very soon. I don't know from where, which country. Many countries are claiming that they are very close to development of vaccine. So I assume that it will be available very soon. And I do hope that it will be available at a cost which poor Indians can afford. That's a very important thing, that it should reach to the billions of the poor people in our country. And the problem is that even if the vaccine is developed and we are immune, it will take a longer time to heal the social relationship disruptions that we have had. So in this process, I would request you to consider involving social scientists in the process. Finally, the present pandemic exposed our unpreparedness. And it is certainly not going to be the last pandemic in the history of humans. So we need to develop a strategy to tackle such possibilities in the future. I wish this webinar a grand success, and I laud the efforts of the organizers from my core of my heart. I hope that all will stay safe and healthy, and together we will overcome this crisis. Thank you very much for your kind invitation.
Thank you, sir.
I would now like to request Dr. Himadri Acharya to deliver the vote of thanks.
Good morning to all of you. Respected Chief Patron of the International Webinar, Honorable Vice Chancellor Professor Dilip Chandra Nath, respected guest of honor, former Vice Chancellor Professor Somnath Dasgupta, respected Patron, Dean School of Physical Sciences, Professor C.R. Bhattacharjee, Head Department of Chemistry, Professor Mondal, conveners of the webinar,
Sound clear. No problem.
conveners of the webinar, Dr. D. Sengupta and Dr. Sudip Chaudhary. Is it clear? Audible? Hello? Hello?
Please
Hello?
continue, sir.
Clear.
Sir, please continue.
Yes. Thank you. Uh, uh, Respected invited guests, all the participants, my esteemed colleagues, staffs, and dear students. First of all, I feel privileged to propose a vote of thanks on this inaugural session. I, on behalf of the Department of Chemistry and on my own, convey hearty thanks to our Honorable Vice Chancellor for gracing the inaugural ceremony and sharing his vision with us for need of such webinar in the current COVID-19 pandemic and need of physical, economical, social and educational measures to sustain with the current pandemic. He also shed light on how contagious animals are becoming threat to the human life. I wish to express our profuse thanks to our chief guest, Professor Somnath Dasgupta, our former Vice Chancellor, for accepting our invitation and agreeing to address participants his experience on the relevance of such webinar in the current pandemic and significance of scientific research to battle with current and future pandemics. He also shared with us how COVID-19 changes its behavior continuously and healthcare sector faces difficulties to tackle the pandemic. I convey thanks to our Dean, Professor C.R. Bhattacharjee, for sharing a brief outlook about the department and his view about various deadly viruses, significance of antiviral drugs, and their discovery to fight against infectious diseases. I would like to thank Chairman of the webinar for his highlight of the department and scope of the webinar for the scientific fraternity. In the current SARS-CoV-2 pandemic situation, we experienced how important is the drug discovery, as most of the antiviral drugs are target-specific viruses to inhibit the virus reproduction and fight against viruses in our body. The potential drug discovery for clinical application in present and future pandemic is a prime focus. In particular, the hunt for antiviral drugs or vaccines to battle with SARS-CoV-2 like highly contagious novel viral diseases. I'd like to express our heartfelt thanks to our national and international speakers for accepting our invitation to shed more light on the drug discovery. We are thankful to Dr. Aviva Levina for accepting our invitation to deliver a talk on transferrin cycle and antiviral drug discovery as a thematic webinar topic. We are also thankful to Professor Ritu Gao for her consent to speak on maturation inhibitors or for antiviral drug discovery. We are thankful to Dr. Giacomo Reina for agreeing to deliver a talk on the importance of nanomaterials during pandemic. We express our deepest thanks to Professor Ayushman Sen for accepting our invitation to give a talk on enzyme motors and pumps. I must appreciate the immense efforts made by the organizing committee to compile a technical program of this magnitude. Organizing such a webinar requires contribution from all quarters. I'd like to thank the technical support extended by the Computer Center, Assam University for convening this webinar. Finally, I take this opportunity to extend our most sincere thanks to all our guest invitees and participants who have joined in this online platform from different places of the country and outside the country to attend the webinar organized by the Department of Chemistry at Assam University to commemorate Silver Jubilee year. Thank you all. Thank you. There was hello, Dibdulal.
Now we will proceed with the technical session one. I would like to invite Dr. Sudeep Chaudhary to moderate the session.
Thank you so much. Now
Okay,
we are
thank you.
heading
I'm
back.
leaving, Professor Mandal.
Okay,
I'm
thank
leaving.
you, sir. Very much. Sir.
Some problem of audio. Okay.
Okay, sir.
So now we are at the technical session 1, and I welcome all the participants and all our guest speakers at the technical session 1. In this technical session, we have 2 speakers, Professor Aviva Lavina from the University of Sydney, and as the second speaker, we'll have Professor Ritu Gaur from South Asian University. This session will be moderated by Professor C.R. Bhattacharjee, who is the Dean of School of Physical Sciences and also faculty of Department of Chemistry. So before initiation of this session, it's my privilege to introduce our speaker, Dr. Aviva Lavina. Dr. Lavina obtained her PhD from Riga Technical University, Latvia, and after a postdoctoral fellowship with Dr. Jacques Mozart in the University of Reims, France, he joined the group of Professor Peter Lay in the School of Chemistry, University of Sydney, Australia, and she is a senior research associate since then there. Her main research interests are mechanistic studies of biological activities of transition metal complexes and the role of metal ions in protein-protein interaction, which are very much important for studying the mechanism and of the different materials and their biological roles. Dr. Lavina has more than 90 papers in reputed peer-reviewed journals with more than 10 book chapters. Today, she will discuss on a topic that is application of bilayer interferometry to the study of transferrin cycle and its application to antiviral drug discovery. In fact, they are the pioneer in the use of bilayer interferometry process to create this cell-free working model of the transferrin cycle. So we believe that we'll get highly motivated, highly enlightened on this very advanced topic, and this will help audience from different prospects, chemistry, physics, medicine, and all other different multidisciplinary segments will get benefited from this. The session will be moderated by Professor C.R. Bhattacharjee. Over to C.R. Bhattacharjee, please.
Just a moment. Dr. Aviva, can you please switch on the presentation mode, please? Please click on the presentation mode. Yes, that will be fine. Thank you. Please continue, sir.
Okay. To have amongst us Dr. Aviva Lavina, University of Sydney, as the first one. He should be speaking on interferometry transferrin cycle and to antiviral drug discovery. In fact, as Dr. Sudeep has mentioned, that she has been a pioneer in the use of bilayer interferometry technique to create the first full cell-free working model of transferrin cycle. She has a vast experience in this particular field. I have pleasure in inviting her to present her talk. Dr. Aviva Levina, please.
Hello, everyone. I would like to thank the organizers for the opportunity to share with you my favorite project, which I have been doing on and off for 7 years now. And recently it has become more topical in relation to antiviral drug discovery. But I will get to that a bit later. So first, I would like to start with the transference cycle. I first read about the transference cycle about 8 years ago, and I was stunned by its elegance. So most mammalian cells obtain their essential iron by the means of a transference cycle. Transferrin is the main iron transport protein in the blood, which gets loaded with 2 iron ions. And the iron-loaded transferrin obtains the right conformation to bind to the transferrin receptor on the cell surface. That this protein assembly is then taken into the cell within an endosome, and within the cell, acidification of the endosome results in the release of iron, which is then included into mitochondrial enzymes or stored in ferritin. But crucially, at the endosomal pH, which is believed to be about 5.6, the 2 proteins stay together. But when they are returned to the cell surface at pH 7.4, the 2 proteins immediately fall apart, and upper transferrin is ready to pick up 2 more iron ions, and the whole cycle is repeated. I have to emphasize that the nature had to invent this extraordinary mechanism, not because there is no easier way to obtain iron, but because cells have to be protected from iron toxicity. As you know, low molecular weight iron generates free radicals and oxidative damage. So we need to make sure that all the iron is bound to proteins while it's processed by the cell. There are specialized cells in our body, particularly in intestinal or lung epithelium, which can take up low molecular weight iron from food or from the air and deliver it to the blood bound to transferrin. But this mechanism is much less understood, so I won't talk about it. Transferrin is a very well-studied protein. It's well known that it consists of 2 nearly equal lobes termed C and N lobes. Each lobe binds an iron 3 ion with 1 aspartate, 1 histidine, 2 tyrosines, and a synergistic carbonate. And it's also known that transferrin receptor forms a dimer on the cell surface, and it binds 2 transferrin molecules. So my main interest in transferrin cycle is to see what happens when iron is replaced with another metal. There is a lot of interest in bioenergetic community in the role of transferrin in transporting medicinal or toxic metal ions. It is well known that naturally transferrin in the blood is only 30% saturated with iron, so there is plenty of room to bind other metals. And it's also known that trivalent metal ions or tetravalent like titanium can bind very tightly to the iron binding sites of transferrin. But our group and another group in the US have argued for years that this mechanism is not doesn't serve to deliver different metal ions to the cell, but on the contrary, it prevents metals from entering the cell because different metals don't bind to transferrin in the same way as iron does, as shown by a few available crystal structures. And in our recent paper in inorganic chemistry, we have discussed the mechanism, how exactly binding of non-iron metals to transferrin prevents their cellular entry. But this is a rather specialized topic in bioinorganic chemistry, which I don't want to discuss in more details. I can come back to this if there is interest. What I wanted to show you is my favorite technique, which is a bilayer interferometry. This is a relatively recent technique to study protein-protein interactions, which has some similarities to a more familiar technique, surface plasmon resonance, but it's much simpler, and the basic instrument called Blitz is very small and relatively inexpensive, which is a advantage. And the principle of its work is remarkably simple. We have a piece of optical fiber which has a chemically modified tip to which we can bind the first protein or biomolecule of interest. And the second biomolecule is in solution, and which can be placed into this tiny drop holder, only 4 microliters. And when the second macromolecule binds to the first one, this changes optical properties of the surface, and this is registered as a change in wavelengths of light. So we get a binding curve. So if we compare with surface plasmon resonance, the most obvious, it's much smaller. And but also equally importantly, bilayer interferometry, unlike surface plasmon resonance, is not a flow technique. And this means that we can change pH and other characteristics of our media in a second. And this becomes really important when we study endosomal processes like the transference cycle, to which I'll come later. But admittedly, it has also a disadvantage. Bilayer interferometry is much less sensitive to smaller biomolecules. So you cannot study, for example, drug binding to your protein. We were fortunate to come across this technique and realize that it can be used in the studies of transference cycle. So we used this to create the world's first functional cell-free model of transference cycle. And this was done by using a BLI probe, which has nickel and Ta bound to it. This is a common tool in molecular biology to capture his-tagged proteins. And we bought a his-tagged human transferrin receptor from commercial stores, and we bound it to that probe. So we are now looking at the probe, which is already loaded with transferrin receptor. And if you look at my simplified scheme of the transferrin cycle, so in the first step, we bind iron transferrin, which is also a cheap commercial protein, to the probe, and we get the binding, which corresponds to binding of iron transferrin to the receptor on the cell surface. Then we replace the solution of transferrin with a buffer alone, that's pH 7.4 buffer. This is a common way to see how tightly the protein binds. So this corresponds to the second step, when iron transferrin together with the receptor is taken into the endosome. And then crucially, we replace the pH 7.4 buffer with pH 5.6 in the presence of citrate, which we believe mimics the endosomal condition. So that's the acidification step of the endosome. So this should result in release of iron from transferrin, but we don't see it in biolayer interferometry because it's not. It's only a small molecule and it doesn't change the surface properties. But when we return pH to 7.4, we immediately see the proteins falling apart, and that's exactly what happens in the transferrin cycle. And also importantly, we regenerate the probe, but transferrin receptor remains bound. So we can use the same probe all over and over again for at least 25 cycles to repeat that transferrin cycle. So that we published 4 papers so far on using the system to the studies of transferrin cycles. So I'll go through what we did with this system. First, we wanted to see if our model reproduces the known features of transferrin cycle. And we saw that human transferrin receptor is very selective for binding human transferrin. Bovine transferrin doesn't bind at all, and despite quite small differences in the protein sequence. So that really shows you the power of selective protein-protein interactions. And second, we saw that replacement of natural carbonate with oxalate prevents the iron release from the transferrin receptor assembly. So when we return to post-endosomal pH, there's no proteins don't dissociate, and that's what has been described before us by a different group. And there are also some speculations in the literature that this may be one of the contributors to autism, because in autistic people known to have higher oxalate concentrations in the blood. Then I spent a lot of time modeling the thermodynamics and kinetics of that process. So it took me some quite sophisticated kinetic modeling, but the result of it was basically that I could reproduce the known kinetics and thermodynamics of transferrin cycle in very good agreement with the literature, and also that confirms that we see binding of 2 transferrins to transferrin receptor dimer. So that's all good, but that doesn't add much new to the existing knowledge of transferrin cycle. What was really new in our work is the exploration of endosomal conditions for which is not easily done by either cell-based techniques or by surface plasmon resonance. So again, we bind iron transferrin to the transferrin receptor, then we wash the probe with a buffer, then we apply various endosomal conditions, and after that we see if iron was released by changing pH to 7.4. If we don't see the release of iron, we do a regeneration step under the conditions which are known to release iron, and then we can reuse the probe. So we found that physiological concentration of citrate 0.10 millimolar is very, at pH 5.6, is very efficient in removal of iron from transferrin receptor assembly, and it becomes even more efficient in the presence of ascorbate. And it's known from nutritional studies that citrate and ascorbate act in conjunction to improve iron metabolism. So that came very well together. And the chemical explanation for this process, we believe, is that citrate, being a stronger acid, replaces carbonate under endosomal pH and binds to iron transferrin, which leads to partially open transferrin conformation. And ascorbate then comes and reduces iron 3 to iron 2. So that iron 2 is no longer strongly bound to transferrin, so it's released in the low molecular weight form, and it can be exported from the endosome through divalent metal transporter. So this is a very known iron chemistry, but we applied it to a biological system, and we were able to publish it in a respected journal. And I was very glad recently to see that there was an independent confirmation that the environment in transferrin receptor associated endosomes is indeed strongly reducing. Because transferrin receptor is the best studied endosomal process, people used it to explore the endosomes by using this sophisticated fluorescent dye that has a sulfur-sulfur bond connecting two fluorophores, and a short peptide sequence which can be linked to transferrin receptor on the cell surface by this plant enzyme called butylase. So the fluorescently tagged transferrin receptor is then taken inside the cell, and because the environment in the endosomes is reducing, the sulfur-sulfur bond is cleaved and the fluorescence is released. So yes, that confirms our theory that ascorbate as a strong reductant is really important in iron release in the endosomes. And now I can finally come to the virus. When I was studying transferrin cycle, I could not miss the fact that it's increasingly talked about in relation to viral infections. Viruses are well known to hijack receptor-mediated endocytosis to enter the cells, and there is a long list of viruses which are known to use transferrin receptor to enter cells. Perhaps the New World arenaviruses are known to use it as their main entry gate to the cell, but HIV and hepatitis C are able to use it as one of the possible routes. And the using of transferrin receptor is particularly advantageous for the viruses because transferrin receptor is designed to be returned to the cell surface. Not all receptors do this. So there is a possibility for super infection. It appears that the coronavirus that we are all talking about is not using this route. It uses another receptor on the lung cell surface called ACE2. But the mechanism by which it enters the cell is essentially the same. It's binding to the receptor, followed by clathrin-mediated endocytosis. So there is a obvious similarity to the transferrin cycle. And if we look at the structure of transferrin cycle, it's interesting. It has three main extracellular domains, and all the transferrin and another protein that associated with transferrin cycle called hemochromatosis factor bind to protease-like and helical domain, sort of to the sides of transferrin receptor. But that leaves the apical domain, the one which on the front, free to bind other particles. And it seems that virus particles are binding to that apical domain, as shown here. And interestingly, that site is also binds ferritin. It's very unusual that two very different mechanisms of iron delivery share the same receptor, but that gives us an advantage because ferritin is a readily available protein, and we can use it in place of a virus to model this process. And also, not only viruses are using transferrin cycle, but also the malaria parasite. And the main cell recognition protein of malarial parasite is known to bind to transferrin receptor synergistically with transferrin. So there's a lot of structural information becomes available, and that inspired some scientists to develop possible antiviral drugs. So at the beginning of this year, there was a publication from an Israeli group that engineered this transferrin receptor analog. This was done to fight New World arenaviruses, which originate from rodent species in South America, but they can become lethal to humans. So this engineered protein is a model of rodent transferrin receptor, which binds the arenaviruses stronger than human receptor does. So it acts as a decoy for viruses not to bind to the cells, but to bind to that protein. And furthermore, they attached an antibody to their engineered protein, so the viruses can be better recognized by immune system. So it's a very sophisticated approach, and it worked well in animal models. But the question is, how much will this all cost? And certainly not a solution for a pandemic like this. So that's this inspired us to seek a collaboration with a researcher in our university, Dr. Tobi Pasiaura. He spent some years in Japanese group which pioneered the system of screening unusual cyclic peptides for drug purposes. And their system uses engineered RNA to create billions of these cyclic peptides and then to determine which of them can bind to the target protein. And they also already had some success in finding drugs that prevent the entry of hepatitis B virus through receptors that are originally made for entry of cholesterol without disrupting the entry of cholesterol. So we wrote a grant proposal to apply the system to screen for drugs that can prevent viruses binding to transferrin receptor. And that was a few months before the pandemic started. And I already used biolayer interferometry to test the potential, just a trial system to test the potential drug candidates using the fact that ferritin can replace the viruses in this system. But then we obviously switched our attention to the coronavirus, and we thought if our BLI system can be useful in looking for drugs against the coronavirus. There are already some data in the literature which use biolayer interferometry to measure the binding of coronavirus spike protein, that famous trimer, to ACE, to receptors. And they showed that the second SARS, so the current coronavirus, binds to the receptor stronger than the first SARS virus. And I obtained the spike protein and the ACE2 protein from collaborators in our university who are also working on this antiviral drug discovery. And I was able to reproduce these results quite easily. So that's not a big problem. So, but what we really want to do is to go further and use the advantages of BLI to study the endosomal conditions. So to see if we can not only can prevent binding of the virus to the cell surface, but also prevent its release in within the endosome. And that's something hardly been ever looked at. There is not much understanding of what happens with the virus in the endosome. But I've started the experiments already, but it's too early to talk about the results. So to summarize, I've got a lot of pleasure from working with BLI on transference cycle, and it remains to be seen. Hopefully something good will come out of the virus project. I would like to thank my mentor, Professor Peter Lay, who was also Devashish's supervisor in his PhD. And we're fortunate to have this wonderful facility in our university that, and it's had Dr. Donna Lai, who had the insight to buy a biolayer interferometry machine, which wasn't very well known at the time, and also Australian Research Council for funding. And thank you for your attention.
Thank you, Dr. Aviva. It was a wonderful presentation. We could know actually many new things about the technique that you have so proficiently used in the interpretation of the transferrin molecules and the target interactions. So please stay with us for some discussions. We have some queries from the participants. I will put it to you one by one. Okay, ma'am? Okay, fine. The first query that I have received is biolayer interferometry useful for antiviral drug protein interactions? Are there any limitations?
Yes, it's certainly useful to study any protein-protein interactions as long as you have recombinant proteins to work with. It doesn't work with actual cells. And the related limitation is that recombinant proteins, usually people try to make them water soluble. So these are active domains. Typically, if we talk about receptors, we try to make an active domain, which is on the outside of cell, and that doesn't involve the membrane component. And membranes are very important in all these interactions, so it's not so easy to model with biolayer interferometry. And yes, so that's it is looking at proteins a bit in isolation from their natural cellular environment, but it's certainly a good start. With transferrin receptor, we were incredibly lucky that the extracellular part of transferrin receptor does all the job, and our system reproduces the natural cycle very well. It may not be the case with other systems.
Thank you. There is a question on actually people are not very familiar with biolayer interferometry. If you can just give its sort of principle in 1 or 2 lines for people to understand this new approach, what you call BLI.
Yes, I already trying to do this. It's simply measuring light interference with the surface of optical probe, and that interference changes by binding of large biomolecules to that probe. So that results in the shift of light wavelengths of light. So I cannot explain that.
Yeah, I understand. Yeah.
What we see as a result is a curve protein binding or dissociation.
Okay. I have another question from the participant. Why endosomal iron release from Fe2 transferring R1 adapts is faster in presence of citrate and ascorbate, concentration ratio 1:10, than their individual presence? What are the role of?
Yes, so we believe so. That's the mechanism. Yes, regarding the role of ascorbate, we believe that it's this reaction here. So it reduces first citrate binds to iron transferrin, and ascorbate reduces, that helps in reducing iron 3 to iron 2, and ascorbate is also known to be a complexant of iron. So that all citrate and ascorbate act in synergy to remove iron from the protein. The question about sodium and potassium, it's known that potassium ions are much more abundant in cells compared with sodium, which is opposite to extracellular environment. So the belief is that endosomes have a particularly higher potassium concentrations, and it has been shown in the in vitro experiments that higher potassium concentrations assist the removal of iron from transferrin receptor assembly. I don't think anybody knows why, and I don't think anybody knows what's the real environment in endosomes is. We are very limited in our means to know what is in the endosomes. We can only use these fluorescent probes at the moment, and they often interfere with, so they may not always give us objective picture because they change cellular metabolism. But the belief is that higher potassium is important in this. Yes, I sorry, I cannot hear you. Your microphone is muted.
Yeah. What is the other alternative for people suffering from autism?
Well, it's still a very controversial topic whether oxalate has really something to do with autism, because that the paper which I am referring to actually showed that oxalate is unlikely under the conditions of blood, even if there is increased concentration of oxalate, it cannot replace carbonate in transferrin, because there is always 25 millimolar carbonate in our blood, and whatever high oxalate concentration is unlikely to do this. So there is no, and not at the moment, there is not an established link between oxalate and autism. Recommend any treatment based on that. I know that people are looking at the links between nutrition and autism, but it's not to go into that.
Okay, ma'am. We don't have any more questions now. Thank you, ma'am, for a wonderful presentation on a very frontier topic. And we are very grateful for your presentations and being with us in the technical session 1.
Thank you.
Apprising us about the latest developments in your subject, and it's very relevant in the current context. Thank you, ma'am, once again. On behalf of the organizers and as a moderator, I thank you for your wonderful talk. So much of knowledge was there. Thank you, ma'am.
Thank you.
Over to Shudip, Dr. Shudip.
Thank you, Professor Lavina, for the wonderful discussion and interaction. We have our second speaker for this session, Gaur, University. Professor Gaur did her University New Delhi and a PhD from National Institute of Immunology. Gaur has served as a fellow award for research in the to continue moderation.
Thank you, Dr. Shudip. Welcome, Professor Ritu Gaur from the Faculty of Life Science and Biotechnology, South Asian University, New Delhi. She will be speaking on antiviral drug discovery, maturation, inhibitors, a novel class of HIV antiviral. She has a vast experience in this particular field, and I'm sure we will be able to benefit from our presentation profusely in this technical session 1. So I now take this privilege to invite Professor Ritu Gaur to present your talk, please.
Very good morning to everyone. I hope I'm audible to all.
Yes, you are audible, ma'am.
Okay, thank you. So I'll start by, first of all, by congratulating the Department of Chemistry. Silver Jubilee. It's indeed a very proud moment. I would like to thank the organizers for organizing this wonderful webinar with a very apt theme. In particular, Dr. Debashish, with whom we are collaborating, for quite some time now. So the title of my talk is Maturation Inhibitors, a novel class of HIV antivirals. So this is something I've been working on for the last almost, 15, 20 years. So I was informed that the audience consists of a lot of students. So, I'm going to talk a bit about what are viruses, and I'll move on work on maturation inhibitors. And towards the end, I'll focus on, the pandemics which have happened in the world so far, including the current COVID pandemic and the therapeutics being developed against it. So, okay, let me see. I'm not able to. Okay, so viruses are a very unique group of biological entities known to infect every type of cell including bacteria, algae, fungi, protozoa, plants, animals. They infect all organisms on Earth. A virus basically consists of a nucleic acid genome, as can be seen in this figure on the left. It is a DNA or an RNA, and surrounded by a protein coat, which is the capsid, which encloses the genome. And in some viruses, there is an additional lipid bilayer surrounding the capsid, which is the envelope. So viruses are obligatory parasites. They replicate inside the host. They use the host metabolic machinery to form a pool of components when assembles to form the virions. And viruses, like bacteria, they cannot be grown on sterile media, but they require the presence of host cycle. So virus genome has all the information required to complete an infectious cycle. So fundamentally, a virus is just a package of genetic information which is protected by a protein shell. And this protein shell helps in the delivery of the genome inside the host, where the genome is then expressed and replicated. So just let me just tell you in the context of a red blood cell, a bacteria is very tiny. You can see on the left topmost, and then in the context of a bacteria, the viruses are even much smaller. So the viruses range from 20 to around 500 nanomolar at the height, the largest size in diameter. And a virus, as if you large picture, it is very, very tiny particles, and such tiny particles have caused such massive havoc in the disease. There's a big debate in the field of virology whether viruses are living or non-living. As a virologist myself, I would like to believe that viruses are living. They are so deadly, but viruses are considered non-living by most biologists, and this is because they are not cells by themselves. They don't fulfill the definition of the cell. They don't grow or respond. They cannot make their own food, and they don't respond to stimuli. That's the typical definitions of a living organisms. So a virus can only multiply inside a living cell, and outside the cell it is nearly dead organism. But such a small, tiny organism, which is non-living, has caused such great pandemics of in the history of humankind. We all know about the smallpox, measles, pandemic, the human influenza pandemic, the HIV pandemic. So you can say more than 90% of all the viral diseases, of all the human diseases, are caused by viruses. So that is the extent of devastation that a virus causes to humankind. So in general, if we look at the life cycle of a virus, it attaches to a particular host cell, and then once the virus is inside, releases the genetic material where it is copied and translated into viral proteins, and then the virus particles assemble, and then and they are released from the cell. This is the general life cycle of a virus particle. So knowing now a few background about virus and what does it look like, and what is the importance of viruses, study of viruses in the life cycle, let's move on to learn about antivirals. And we know that vaccines can prevent diseases, but vaccines don't have much effect if an individual is already infected, with the exception of the rabies vaccine. So our second line of defense is the antivirals. And antivirals can stop infection once it has started. So, but despite 50 years of research, our arsenal of antivirals remains very small. So we have close to 100 antivirals available in the market today, and nearly half of them are actually against HIV. And then the remaining are against hepatitis, influenza, human simplex virus, HSV, HPV. The number of the antiviral drugs compared to the antibiotics remain very low. So if we look at why there are so few antivirals and why is it so difficult, to develop antivirals. Basically, as we have learned today, that viruses, require a host to multiply. So if we develop compounds which interfere with the virus growth, they can adversely affect the host too. So there are a lot of side effects as the virus engages, host in all the steps. Also, many medically important viruses are very dangerous. They can't be tested in model systems or propagated even. It's very difficult or impossible to grow in an in vivo model. Even in vitro culture labs, culturing of certain viruses require labs, which are not readily available throughout the world. So that's why there are very few antivirals, or the effort has been put on developing very few antivirals. Now, if you just look at broadly what all steps the virus targets in the antivirals target in the virus life cycle, they can affect the attachment of the virus particle to the cell, the uncoating of the virus, the replication of the viruses. There have been certain lead compounds, as you can see in the table, and the viruses against which they have been developed. So there are several antiviral drugs which target different steps in the virus life cycle. So I'm going to be talking about a specific class of antiviral drugs, a new class of antiviral drugs which we are working on, which are called the maturation inhibitors. If we look at the global of HIV, HIV is a retrovirus. It's the causative agent of AIDS. There are 9 subtypes of HIV, A to K. And if on a global scale, the most prevalent HIV genetic forms are subtype A, B, and C, with subtype C accounting for almost 50% of all HIV infections worldwide. So subtype B is the main genetic form in the Western world, the Americas, the Europe, even Australia, whereas subtype C is predominant in those countries like the Africa, the Indian subcontinent, and these countries carry nearly 80% of the global HIV burden. So as you can see in the pie chart on the lower bottom left, it is the subtype C which is the most dangerous, and it causes nearly 50% of the total HIV infections worldwide. So, it is very important to work type C, and most of my lab that we work on HIV subtype C. Just broadly, if you look at the HIV structure, it is roughly spherical. There are 2 copies of RNA. It codes for 9 genes, and 3 of them are structural proteins and 6 accessory proteins. And the virus life cycle involves attachment of the virus to the CD4 receptors, followed by uncoating step, and the viral genome is released inside the cell. So it's a reverse transcription happens. That's the first step, and then to form from the RNA to a cDNA, and then this cDNA enters the nucleus where it integrates into the chromosomal DNA, and then it uses the host RNA polymerases to make the RNA. The RNAs are transported out, translated host ribosome to form all the viral proteins, and the assembly and the packaging of the virus occurs at the plasma membrane, and the virus pinches off and buds off from the membrane. So when the virus is released from the membrane, it is an immature, non-infectious virus particle, and then soon after the release, the viral protease cleaves the gag proteins to make it into a mature infectious form. And it is this step that the maturation inhibitors, which we are working on, which they inhibit this step. So if you inhibit this step, you do not make an infectious virus particle, and the virus released from the cell remains non-infectious. So, there are different targets for intervention which are being developed. We have 6 classes of antiretroviral drugs available. You have drugs available which will affect the entry of the virus. You have CCR5 inhibitors, fusion drugs which prevent the virus from replication, the nucleoside analogs. Then we have integrase inhibitor, which prevent the viral DNA from integrating. Then we have drugs which are called protease inhibitors, which will prevent the uh, protease action so that it does not make a mature particle. And a novel distinct class of drugs, which are the maturation inhibitors, which prevents the maturation of the virus by binding to GAG itself. So that has been the focus of my lab, and we are working on different aspects of HIV biology. We are attacking the virus as well as the host. So we are working on characterizing inhibitors which will affect or inhibit the entry, the maturation, virus release. We are also working on viral latency. That is again a big problem because HIV tends to remain latent, and that's how cells. We are also altering or focusing on altering the host and working on identifying novel restriction factors which can inhibit HIV replication, and also how ion channels and microRNA play a role in HIV replication. But today I'm going to just focus on maturation inhibitors and tell you what work we have done in the past regarding that. So why there is a need to continuously develop new antivirals against HIV? Because of viral resistance. So it's very easy to develop viral resistance to antiretroviral drugs. And this is due to the error-prone reverse transcriptase. So the enzyme reverse transcriptase is error-prone, does not have a proofreading mechanism. So it is very easy to incorporate mutations. And whenever we are giving a patient, it's like adding selection pressure. And when a virus is under selection pressure, it tries to replicate under pressure and tries to evolve under pressure by incorporating mutations. And especially such kind of mutations occur when the replication is not completely blocked. Or there are some monotherapies given, if only single therapy is given, or people do not take the medicines properly, or the drug has other pharmacokinetics problem. So all these lead to development of resistance against these drugs, and that is why there is a continuous need to identify more drugs and to give these drugs. HIV drugs are always given in a combination rather than as a monotherapy. So what is HIV maturation? HIV maturation, as we just said, it is converting the immature HIV particle in which the GAG forms a sphere around the virus, a mature form in which you can see a condensed conical core. And this is the form which is infectious particle. And this is very clearly visible in the electron microscopy picture also. On the left is the immature particle, and on the right is the mature with a conical core in the center, you can see. So how does this maturation is a very highly ordered process, and in which this HIV GAG protein is cut at different places by the HIV protease. It's a sequential step. So the first cleavage occurs before the nucleocapsid, then followed by cleavages which occur between matrix, MA, and the CA, and then the other proteins to release matrix, capsid, nucleocapsid, and the P6. That's what the entire HIV structural, it forms the structure of the HIV particle. So what maturation inhibitors inhibit is this particular step, which cleaves the capsid from the spacer peptide 1. So this spacer peptide 1 is not released, and because of that, the capsid remains attached to the spacer peptide 1, and that is what leads to the aberrant morphology of the virus particle. So this is what we are working on. There are two classes of maturation inhibitors which my lab works on. The first class is Bevribat. It's a dimethyl succinyl derivative of Bevribat, and this Another class of drug is the pyridone derivatives, which I'm not going to be talking about today. So the this triterpene derived from betulinic acid is further derivatized with dimethylsuccinyl moiety. So, one of the, so it was seen that this particular compound in vitro studies showed that all HIV isolates, HIV-1 isolates, and it retains activity against viruses which have developed resistance against the known inhibitors. So that is again very important. It is low toxicity, orally bioavailable, and this is a study we are doing in in collaboration with Panaforce Pharmaceuticals USA, which is now overtaken by another company called DFH Pharma. So if the capsid and the spacer peptide is not cleaved by the maturation inhibitors, it leads to formation. So it remains joined together, and that's why so it becomes a 25 kD protein instead of a 24 kD protein. And that is what we see, that in the presence of Bevrimat, BVM, we see this band, which is a 25 kD band. And this is, as I said, because the CASP1 remains joined together, and this is kD. If it gets cleaved, it forms this 24 kD capsid. So this was very, this work was published in early 2003. We published the mechanism of how these maturation inhibitors act, and it was a very exciting time. And in 2005, the clinical trials of this compound started, and it was given at different doses. It inhibited the replication of the virus, was completely inhibited in the presence of the compound. So I have already shown that in the electron microscope, you see this aberrant morphology of this compound, and because it prevents the cleavage between capsid and SP1. So it was given as a monotherapy during the trials. Patients were receiving 1 dose per day. But what was the outcome of the trial was that only 50% with a drop in the viral load to nearly 1 log, and the remaining 50% were resistant to this compound. So that is why the trial was discontinued, and then on sequencing the genome in the patients who did not respond to the drug, it was found that these patients carry viruses which had polymorphisms in the region the drug acts. So when we sequenced the viral genome, we saw that the SP1 peptide carries certain polymorphisms, especially in this QVT motif. And it is these polymorphisms which prevent the maturation inhibitors from acting. So we looked at the inherent presence of these polymorphism across different HIV subtypes. So we looked for these sequences, changes in various subtypes, across A, D, E, all the subtypes, and we saw that these polymorphisms are inherently present in subtype C. So in subtype C, instead of QVT, there is QAN. So, that is, so maybe these patients, carried the subtype C virus, and that is why they were resistant to this compound. And we did confirm this finding, and when we checked the activity of Bevrimat against HIV subtype C viruses, so NL4-3 is a subtype B virus, whereas the other three, ZM247, INDC1, and K3016, these are HIV subtype C viruses. They are originated from South Africa. INDC1 is from India. So this is just showing that all these viruses carry the QAN motif instead of the QVT motif, which is the subtype B virus. And we saw that, yes, none of these viruses, they were all resistant to Bevrimat. So it did confirm that this could be a possible reason why the clinical trials failed. And, the subtype C viruses are resistant to this compound. So, so it brought back to a drawing board, and our chemist collaborators at DFH Pharma, Dr. Carl Wild, they synthesized a number of analogs of this parent compound Bevrimad by attaching different side chains at the C28 position, of this scaffold. So they attached esters, amines, alcohol, different kinds of groups to it. And they gave us these new compounds for testing against our subtype C viruses. And what we observed was that, yes, the derivatization of these of Bevrimat made them more active against subtype C viruses, and in all the cases, we saw nearly 60 to 70% activity. So the new BVM analogs were active against now the subtype C viruses. This was very exciting, and we did report this work. And the viruses which were produced in the presence of these analogs were non-infectious. And these analogs affected the maturation of the viruses because we tested them against for a virus entry, and these analogs had no effect on virus entry. So they were very specific. They were blocking only the last step of the virus life cycle. And they had antiviral activity in the low nanomolar range, which was again very, very, very good. So this was very exciting, and we continued our work. And the Carvile, the DFH Pharma, they further derivatized the analog, one of the analogs, and incorporated sulfonyl groups and some other group to them in order to improve their efficacy and lower toxicity. So there were these three compounds, 8611, 8612, and 8613, which were synthesized, and they were given to us again to test against our subtype C clones. So we tested them against subtype C, and we saw that, yes, these analogs are highly active, much more active than the previous ones. They are more active at a lower dose. So the previous group of compounds were more active at 1 micromolar, but these compounds are active even at 10 nanomolar. So K301 saw nearly 60-70% activity. But what was surprising was that when we tested these two viruses, these new second generation analogs against the other two subtype C viruses, we saw lower activity than K3016. So within the subtype C strain, we could detect that the activity of these analogs, it varied between the same subtype, subtype C strain. Okay, so all these subtype C strain had the same QVT or the QAN motif. So that was not the reason why the variation is there in the activity. So activity. So it was reduced effectivity for K3016 virus, whereas the other viruses were less susceptible. The virus was a little more infectious even in the presence of the maturation inhibitors. So we decided to sequence these three viruses again, and then we, in the SP1 region, we saw that these viruses carry another polymorphism. So we have at the position 10 of the SP1 residue a glycine present in K3016, which is a 3 in the other two strains. So it could be that the presence of threonine, is at the position 10 is making these two strains less susceptible to this drug. So we thought we will make mutations, in these strains and see the effect of the of the drugs on the mutant viruses. So we created 3 mutants, so we mutated the glycine to threonine in the K3016, and in the other 2 we mutated the threonine to glycine. And now these mutations were created by site-directed mutagenesis. And once we have these mutants, we confirmed whether now is there any differential activity of the drugs on these mutant viruses. And yes, we saw that in case of K3016, mutating from a G to a T, which is less sensitive, decreased the activity of the second generation inhibitors on this virus. Whereas now mutating the threonine to a glycine in both the other viruses, it increased the activity of the compounds. So polymorphism present at this position 10 also governs the sensitivity of the HIV subtype C towards BVM analogs. So its same was reflected in the infectivity of the viruses produced. Also, the K3, the mutant K3016 had lesser reduction in infectivity as compared to the wild type, whereas now we saw greater reduction in infectivity with the mutations in the other two subtypes C viruses. So this is, this teaches us another lesson that there could be polymorphisms between subtypes, like the QVT motif polymorphism was there between HIV subtype B and subtype C, which gives sensitivity to a particular compound or drugs. But within a particular subtype also, there could be polymorphism. So we are currently testing more compounds and further derivatives, because what we want is that we want to develop an antiviral drug which is active across species, across different strains, and not only it is not a subtype specific drug. So with that, let me, this work we have published in quite a few papers. It was about maturation inhibitors, and the work continues. We have also worked on a group of compounds in collaboration with Dr. Devashish Sengupta. So he is a chemist, a very good chemist, and he's working on porphyrins and fullerenes. So there was a very novel, discovery that they incorporated zinc moiety into the porphyrins as well as the nitro, including the nitrophenyl groups. So these zinc nitroporphyrins, which were synthesized, these gained activity as HIV entry inhibitors and also in cancer photodynamic therapy. So this paper we published last year. So just a brief summary of the paper was that incorporation of zinc ions enhanced the biological activity of the porphyrins against HIV, and replacing the meso-4 methylpyridinium cations with meso-4 nitrophenyl groups contributed strongly to the anti-HIV and cancer PDT. So Dr. Devashish is further, we are working on these, derivatizing it further, and our collaboration continues with him. So let me move from the HIV field and talk in general about pandemics which have happened, in the world, and the current pandemic. So if we look at the history of the viral outbreaks, there have been several major viral outbreaks since the end of the 19th century. And it started in 1918, the century started with 1918 with the Spanish flu pandemic, then followed by, the world has witnessed several other disease outbreaks. For example, the Asian flu, Hong Kong flu, SARS, HIV/AIDS, swine flu, Ebola, MERS, and now COVID-19. So there has been a lot of pandemics which have affected the world. Talking about the influenza pandemic, the influenza pandemic always comes in waves. It started with the 1889. A flu pandemic started in 1889. The famous 1918 Spanish flu. 1957 was the Asian flu. Then 1968 was a Hong Kong flu. 2009 again, another flu pandemic started in Mexico and spread throughout the world, and followed by 2000 many flu epidemics. So the 2000 all these flu, especially the 2009 flu pandemic strain, it's actually a strain which is a reassorted strain. So it's a reassortment of avian, human, and the swine flu. So that means mixing of the genes has happened. And how these mixing of genes happens, it always creates a new strain. So usually the mixing of the genes happens in an organism or inside cells which are susceptible to all the strains. For example, pigs. Pigs, are a great. So pigs infected by the human influenza virus as well as the bird influenza virus. So the mixing of the strains occurs, specifically the envelope proteins, the chemoagglutinin and the neuraminidase proteins. So if a particular cell is infected by both two different strains, so when a new virus is formed, it mixes these proteins from the two different strains to form a new virus, which can cause a new kind of pandemic. So now these viruses can infect both humans as well as birds. So that is how, by incorporating these mutations, new influenza pandemics are, being affecting the world. Let's switch to the current problem we are facing, human coronaviruses. So coronavirus, it's a Latin means crown. So, as you can see on the left, these viruses have these spikes on their surface, which reflects like a crown, a king or queen's crown. So that is why it's called a coronavirus. It is an RNA virus, belongs to the family Coronaviridae, and there are actually 4 major subtypes of these subgroups of the virus: alpha, beta, gamma, and delta. And based on their genome structure, alpha and beta viruses infect only mammals and causes all the respiratory symptoms in humans and gastroenteritis in animals. So until December 2019, there were only 6 different coronaviruses known to infect humans. And these were the HCoV, which I've listed here, 1 to 4, and MERS and SARS. So until December 2019, these were the only 6 coronaviruses which were known to infect. And since December 2019, a new virus has been added, which is 2 virus. So this virus, as you can see in the figure, it is a spherical enveloped particle, positive single-stranded genome. The genome is bound by a nucleocapsid protein. It's surrounded by a membrane protein, and then there is another additional layer, the envelope, which carries the spike proteins. And the spike protein is the primary receptor binding domain, and it is critical for virus entry. That is what it attaches to the host cells using these cellular receptors called the ACE2, which is the angiotensin converting enzyme 2. So the coronaviruses, if we just compare the sequences between identity of the current SARS-CoV-2 virus with the other previous SARS and MERS virus, we see that this spike protein, there is 76% sequence identity with previous SARS-CoV virus in general. But if we specifically compare the receptor binding domain of the 2 SARS-CoV virus, there is more than 95% sequence homology. And that is why, people think that the SARS-CoV-2 also uses the same receptor to gain entry into the virus. So just the coronavirus pandemics, there was a SARS in 2002, 2003. The SARS pandemic caused SARS, which resulted in 10% mortality. Similarly, the MERS pandemic happened in 2012. It had much higher, greater mortality, nearly 37%. And the ongoing coronavirus disease 2019, the COVID-19 disease, has already affected more than 19 million people, killed more than 7 lakh people worldwide. So if we look at the origin of these human coronaviruses, all the viruses initially existed in bats, as cove-related viruses, and then they slowly acquired mutations. For example, the SARS coronavirus acquired mutation to gain entry into an intermediate host. And for this virus, the intermediate host was civets or raccoon dogs. And it further acquired mutations, and now it could infect humans. The MERS virus from bats mutated to gain entry into an intermediate host camel, and now it further, acquired mutations, and now could infect humans. In case of SARS-CoV-2, the origin is that, but we're really not very clear about what is the intermediate host. There are several reports pointing towards pangolins and other animals, but it is still not 100% from these intermediate hosts, and now it has gained infectivity in humans. So that's how, by acquiring mutations, the viruses expand their tropism and the host in which they infect. So in order for any viral disease, to be a pandemic, there is a number called an RO number. So the RO number, the public health experts, they always look for this basic reproduction number. And this number measures the disease potential, and it represents the average number of people who will catch the disease from one infected person. Okay, if in a population which are disease free. So if how many people one person can infect minimum, and the RO number is CoV-2 is around 2 to 3, 2.5. And in order to contain a pandemic, we have to reduce the RO number to below 1. So that is what the health disease experts always would like. To control a pandemic, the RO number has to fall below 1. So the general life cycle of SARS-CoV, it binds to this receptor, or the ACE2 receptors on the surface, gains entry into the cell, followed by release of the viral genome. The viral genome, the first step is translation. It's a positive single-stranded RNA, so it is translated to produce the viral proteins. Some of the proteins are viral polymerases, and then they make more of the virus genome. And now this genome is, along with the viral protein, is packaged. It goes through the ER-Golgi network. It's packaged inside the ER-Golgi and is released out into the cytoplasm. So a lot of research has been going on to study or to inhibit these steps in the virus life cycle. And so just like other viruses, the SARS-CoV-2 also hijacks, you can see the machinery, the host machinery to its advantage, and replicates and translates using the host machinery. So several targets have been developed which can be inhibited to inhibit the virus replication. So ACE2 is one of the critical targets. So, if you can prevent the virus from entering the cell, that is the best possible thing. So there are a number of inhibitors. There are spike protein vaccines which are being generated. There are inhibitors which prevent the, so when the, so ACE2, let me just tell you a little about ACE2. It's a transmembrane it's an enzyme which plays a key role in the renin-angiotensin system in our body, which is basically controls the blood pressure. It's present in the lung cells, but even in the kidney, the GI tract, could be in the brain too also. So you can develop antibodies, which can specifically block virus from attaching to ACE2 receptors. Convalescent sera from patients carries antibodies, and that is could be useful in preventing the attachment of blocking the attachment of the virus to the cells. We can overexpress ACE2 receptors, so you can have a soluble form of ACE2 receptors which will attach to the spike protein, and kind of saturate the spike protein and prevent this from entering into the cell. So any possible number of ways are being developed, which of modifying ACE expression, and so that this entry of the virus is inhibited. Again, another target could be the TMPRSS2. This is basically a protease, and this TMPRS inhibitors have been developed, which prevent SARS-CoV-2 entry into the host cell. So this protease is basically, it's a serine protease, And so for a virus to enter into the host cell, the spike protein has to be cleaved, okay, at two different sites, and this is done by this serine protease. It's a cellular protease. So if we prevent this cleavage, then you don't, you do not get the spike protein from entering into the cell. So camostat is one of the inhibitors. It is being, used to treat currently used to treat chronic pancreatitis, but it is now being tested against this SARS-CoV-2 infection as well. So blocking the entry, then there's a lot of discussion, a lot of growing interest in chloroquine and hydroxychloroquine as potential treatment for COVID. So once the virus enters the host cell, as I said, they are in endosomes, and we have to release the virus genome in the cytoplasm, and this happens by lowering of the pH of the endosomes. And so chloroquine and hydroxychloroquine, they raise the pH of the endosomes, and this prevents the virus entry into the cytosol. There can be other mechanism also. Chloroquine may reduce glycosylation of ACE2 receptors, which is critical for SARS-CoV-2 binding. It might block the production of pro-inflammatory cytokines, therefore, pathway, which may lead to acute respiratory distress syndrome. So upon a lot of possible pathways in which chloroquine and hydroxychloroquine could act, but still at present, considering all the data, there is not sufficient evidence to endorse, giving patients these drugs as safe and effective treatment for COVID-19. Another very popular target against COVID-19 is the RNA polymerase. So this is an RNA-dependent RNA polymerase. It is a viral polymerase specific for the virus and produced by the virus. So there are a lot of drugs which are known, which are being treated for other viruses, but which have been now repurposed for use, in SARS-CoV-2 treatment. Like ribavirin, it is used for hepatitis C virus, respiratory syncytia virus, RSV. Remdesivir initially for hepatitis C and later for Ebola virus. Sofosbuvir, which is used for hepatitis C viruses. Galidesivir, which was again for hepatitis C and later for Ebola and Marburg. And tenofovir, which is used for HIV/AIDS treatment and also for hepatitis B treatment. So all these are basically nucleoside analogs, and they affect the replication or the step in which the RNA-dependent polymerase is involved by either binding to the polymerase and inhibiting the activity of the enzyme, or incorporating in the chain. So a popular among these is currently, you must have all heard of this drug called remdesivir. This is the only FDA approved drug which has been approved by FDA for COVID treatment. It has also been approved in India, and this is, as I said, is a repurposed. This is given as a pro drug. So in a proactive form, and it is taken up by the cells, and it is then converted into an active form. It's the active form is an adenosine triphosphate. So this change occurs using the esterases and the proteases, kinases present in the host kinases. They activate these prodrug into an active form. And once this drug is activated, it is an adenosine triphosphate. So it mimics the normal ATP present in the cell, and it is incorporated into the growing chain instead of the normal ATPs. And so once it is incorporated, then it leads to stalling of the ribosome, and it leads to premature chain termination. So RNA replication stops. So, stop. That is how this drug acts. It's a broad spectrum antiviral, and it has gained a lot of. So still, it is not given to patients only with serious symptoms. So another compound is favipiravir. It is used for emergency use, and it is now available in India also as FabiFlu. Again, it targets the virus replication. So India, the companies Cipla and Hetero are now making generic forms of remdesivir, and they're called Covi-4 and Ciprimi. It is already launched, and it inhibits viral replication. It is still it is used under restricted emergency use drugs, only in case to treat severe cases of SARS-CoV-2. So this is just a list of drugs. I've already talked about quite a few of them. I've talked about ACE2, inhibiting ACE2, TMPRS2, independent RNA polymerase inhibitors. Now there are certain proteases which are also crucial, for conversion the viral polyprotein into enzymes. And there are drugs like lopinavir and some other darunavir which are could be used to treat. So as such, there is no drug or no compound which is specifically developed against COVID. So all the compounds, all the drugs which are being tested or used to treat COVID it's all drug repurposing which is happening right now. And that is why it is very important for drug repurposing. Also, it is important that we should eventually in the long run look for development of broad spectrum antivirals. To prevent that is a lot of research is needed to develop broad spectrum antiviral drugs. So instead of one drug, one virus, so have a specific drug for a particular virus, multiple virus paradigm. So that is we have to discover new broadly acting antiviral drugs. Could be small molecules. So the whole antiviral drug discovery, it's something which takes a long time. You have to start by testing the drugs in vitro cultures, then primary cultures, look at the pharmacokinetics, toxicology studies in animal model. So FDA approval always requires the drug to be tested in 2 different animal models. Look at efficacy, and then we have phase 0, 1, 2, 3, safety, efficacy, large scale trials, and then only you have, so it's a long process, which could take, 10 to 15 years easily. So this is an area where the research should be focused. This is a very, busy slide. This just shows you that researchers all over the world are trying to find the use of these repurposed drugs and affecting different ways, different pathways to prevent COVID replication. So there is a huge effort going on around the world to develop or to stop this pandemic from happening. A lot of different drugs are being tried, which were working for other viruses. Another, class of broadly spectrum antivirals, which are under research right now, are lipid modifying drugs. So these, one important pathway in the viral replication is lipid metabolism. So the virus hijacks all the host machinery, and so virus hijacks this pathway as well to make its own viral membranes. So, you can have inhibitors of virus cell entry, cytosis, as well as inhibitors of some cellular pathways, which are involved in the virus life cycle. So we can have this drug called methyl beta cyclodextrin. It works by depleting cholesterol. You can have direct inhibitors of endocytosis, chloropromazine, chloroquine, I've already talked about. Arbidol, these are direct inhibitors of endocytosis process. So another drug which is under research is the LJ001. So this is a membrane binding compound, very promising antiviral mechanism, and it enters only the viral membrane and not the host membrane. So this is again under research. It was given as a given as a drug against the Ebola epidemic or the HIV influenza, but still it is under trial and not confirmed. Another drug which is the cholesterol. It is an important component of the viral cellular membranes, and statins have been used to reduce cholesterol synthesis by inhibiting the activity of the enzymes. So these potential lipid modifying drugs could be developed in future as broad spectrum antiviral drugs. So considering viruses and their role in pandemics, a lot of effort has to be made in order to develop novel drugs and to look at development of drugs or broadly acting antiviral drugs which can alter or affect different viruses and not virus specific. So let me just summarize by saying that looking at the past and elucidating the role of specific molecules and how they confer virulence and transmissibility, it will teach us a lesson how to deal with the future pandemics. Our advances in the next generation sequencing are improving our knowledge about viruses, and this is helping us design small molecules to specifically target the virus and not affect the host. This will help in reducing side effects of a particular compound. In the long run, we have to think of investments in global partnership. We have to build up laboratory capacity worldwide, improve research on viruses, strengthen surveillance networks, strengthen diagnostic capabilities, and all this will improve the identification of new viruses and try to stop them before they become a pandemic. So we have to be committed. It's had to be a global commitment. Sharing data about virus isolates, supporting innovative research, dedicating resources. All this together will help emerging viruses and control the pandemics. So with that, I would end my talk. And so all of us, when the year began, we had a lot of goals, which were general goals every year, but this year is special. And so managing this pandemic is a major goal. So I wish all of you stay safe, stay healthy. I would like to acknowledge all my students, South Asian University, specifically my students Yuvraj and Madhu, who have helped me in compiling all the slides. We have taken references from multiple articles. There are some of them are listed here, and I acknowledge all of them. And thank you for your attention, and I'll be happy to take questions.
Thank you, ma'am. Illuminating and lucid from your side. The presentation actually not only lucid, but it has actually triggered a lot of questions.
Yes.
The undergraduates whom has participated and joined this particular webinar. So congratulating for a full presentation. It was complete in all respect, giving us an overview of the current pandemics, the drugs, and the research on maturation inhibitors that you have been doing. There are a lot of questions actually which have been received, but I'm not quite sure that I'll be able to take all of them or not. But some diverse types I will take. First, of course, I will start with the one which I have received from the youngest participant, perhaps, who is from an undergraduate college.
Okay.
So I take this question for you to answer. He mentions, why mostly the host of a deadly virus is an animal?
Well, the host of a deadly virus. So yeah. Yes, so so animals do carry a lot of viruses. So animals, so bats are considered the source of more deadly viruses, and so viruses which we did not even know of before. And due to, urbanization and clearing of the forest, the humans came in contact with bats. So there are many animal viruses which do not, we don't even know about them. But then once you come in contact, then the virus has another new host in which, the mutations make it gain, infectivity into those new organisms. So yes, he's right. Animals do carry a lot of viruses, but there are viruses which are human specific as well. So.
Okay, thank you, ma'am. The next question is, why some viruses are non-infectious even though they buds inside the cell?
Okay, so in order for a virus, so viruses are everywhere. So the table we are sitting on, the chair we are sitting on, everything you can say is there are viruses everywhere. But the reason why all the viruses do not cause a disease is because of our immune system. So our immunity, our immune system, takes care of mostly all the infections. So it's when we have a weakened immune system that a particular virus becomes deadly. And also a virus can cause a disease only if a virus is able to infect it. And to infect a particular cell, the virus has to have the right receptor to gain entry. So that is why. It's a combination of having the right environment for the virus to grow and our immunity. It's an interplay between the two, which leads to disease.
Thank you. The next question is, by how many years maturation inhibitors can delay the progression of HIV?
Well, there is, yeah. So there is no specific answer. Maturation inhibitors, when they come into actually when they get FDA approved and come into practice, they will be always be given in a combination therapy. So right now the combination, the treatment, the way treatment is done for HIV, there are 3 drugs which are at least minimum 3 drugs are given. So the protease inhibitor, reverse transcriptase inhibitor, in combination with maybe an entry or any other inhibitor. So whenever maturation inhibitors will always be given in a combination, and so that the virus does not develop resistance. So there is no specific number of years it may last. But yes, maturation inhibitors have expanded the repertoire of the HIV, anti-HIV drugs. So that is how.
Okay. The next question is, how molecular selection is done to
Okay. So this is more in literature. So, like Bevrimat, I can say that Bevrimat, it was a plant product. So it was a betalainic acid, a plant. And so, we have now a lot of different ways in which new drugs are being. So you have chemical libraries available, and people are doing mass screening of the libraries, to identify compounds which have activity. So it starts with a small cell culture based study, and then the ones which show some activity are further scaled up into a larger study. It is basically searching for libraries, looking for compounds which show activity, and that is where you start. Yeah.
Okay. Related to HIV infection, someone wants to know, what is the scope of nano drug delivery system?
Oh yes, nano drugs are the future. And so, the one of the ways how to deliver the drugs, so nanoparticles will definitely help in the delivery of the drug right cells. So yes, it is an exciting future, nano drugs.
Okay. The other question is, as we discover a vaccine which will efficiently inhibit virus replications with the change of genetic modifications.
I'm sorry, your voice. Can you just repeat the question?
Yeah. As we know, most of the viruses are mutants. So how we can discover a vaccine which will efficiently inhibit virus replications?
That is a challenge, yes, and that is yes. So that is a good question. It's that is how, what the challenge is. You know, the HIV virus, for example, it was discovered in 1986, and we still do not have a vaccine developed against it because there are so many. So a person who's infected today, if you isolate the virus from the same person a month later, you will have a different strain, a mutant growing inside the same person. So that is why we have to design vaccines looking at the conserved epitopes, conserved regions, which are not prone to uh, mutations. So that is a challenge, and this is more specific for RNA viruses. So RNA viruses do not have an error-prone RNA polymerases. So the mutation rate is much higher than DNA viruses. That is why it is difficult to design a vaccine. So look for conserved epitopes. That is
Okay, I think this would be the last question, though there are many, but I would appreciate that if the organizers can make some provisions for
yeah, they can
sending
send me the
the
questions.
rest of the questions by email to
Yes.
the to Dr. Professor Gore for her to attend at convenience. So this is the last question that I will take is how can virologists like yourself who work with the safety to handle HIV practically work with more contagious viruses like SARS-CoV-2 and others that have extreme safety concerns or threats to health? Or how should the virologist of the future prepare to handle more contagious forms of viruses?
Yes. So so that is a very important thing, because for a virologist to work on such contagious viruses like SARS-CoV-2, influenza viruses, so you need specialized facilities. You know, we have the biosafety labs 3 and 4, which are needed to work on Ebola and influenza, different strains which are highly contagious and airborne. So these are airborne. So, I mean, for a virologist or not, not to have a BSL-4 facility. So what we can also do is we can make chimeric viruses. So that is our plan, that we are using the backbone of HIV and incorporating the SARS-CoV envelope protein, the spike protein in our HIV backbone, and then the virus will express the SARS-CoV envelope. And then we can use our own model systems, our own cell culture system, the way we do for HIV, and look for inhibitors, of entry and things like that. So chimeric viruses are one option which we are going to try.
Okay, ma'am. So with this, I would like to express my sincere gratitude on behalf of the School of Physical Science as well as the Department of Chemistry for giving me this opportunity to moderate the first technical session today. And from the School of Chemistry, University of Sydney, Australia, for the first talk in this technical session that she delivered on a very important technique, biolayer interferometry, and there were effective interactions also. So I thank you, ma'am, for sharing your time with us and also sharing your advanced research in this very pertinent theme of today. So, and then I come to thank Professor Ritu Gore for her presentation on the antiviral drug discovery, variety of aspects that she touched on. And I really express my gratefulness from the department as well as from the school for your presentations, which was very lucid, and it was well understood by the participants, because many of them are not initiated into this kind of interdisciplinary kind of research pertaining chemistry, biology, physics, theoretical relations, etc. And we have actually extended this scope of this particular university students and scholars. So they have also got a chance to hear your presentations on these advanced theme on antiviral drug discovery. So I thank you, both the speaker, for being with us in this morning session. And I now would like to hand over the hand over this session actually to the organizers. I think Dr. Sudeep will take over, whoever. But before I close this technical session, not really close, before I hand over the presentation scope, I would like to actually express my gratitude to Professor Muhammad Rafiqul Islam, who has been one of the founding head of the Department of Chemistry and retired some years back. And he is also there today with us, sharing the screen. And we are celebrating our 25th year of existence. This is a silver jubilee year of the department. To have Professor Islam to share the screen with us today on this auspicious occasion of our silver jubilee ceremony. So welcome, Professor Islam. Thank you for being with us. So with this, I now give it over to Dr. Sudeep Chaudhary. Thank you, everyone, and also thank all the participants for generating so much of enthusiasm and making it possible that such an intense interaction with the speakers. I thank all the participants from the core of my heart to make this technical session one very, very successful.
Well, thank you, Professor Bhattacharya. Yeah.
Thank you.
Yeah, I would also like to thank Dr. Bhattacharya for running this session as chairperson, and all the organizers for inviting me again. It was very informative. Thank you. Yeah. Thank you, everybody.
Thank you, ma'am. Over to Dr. Sudeep Chaudhary.
Thank you so much. And it was a nice and vibrant session and interaction from both the speakers, and a nice curation of this program by our moderator. Now I request Professor S.K. Jashimuddin to offer a vote of thanks for this session and declare the closure of this session one along with. Over to Dr. Jashimuddin.
Thank you, Dr. Sudeep. Respected moderator, speakers, invited guests, participants, ladies and gentlemen. It's my privilege to have been asked to propose a vote of thanks on this first technical session of the webinar. I, on behalf of the organizing committee, convey my regards and heartily thank Dr. Aviva Levina for her valuable speech on application of biolayer in interferometry to the studies of transferrin cycle and to antiviral drug discovery. I also extend our deep sense of appreciation to Professor Ritu Gaur for her inspiring talk on antiviral drug discovery, maturation inhibitors, a novel class of HIV antiviral. I wish to extend my sincere gratitude to Professor Chiraranjan Bhattacharya, Dean, School of Physical Sciences, for moderating the technical session. I want to thank our ex-professor of the department, Professor Rafiqul Islam, for sharing the screen with us in this webinar today. I thank all the participants for being with us in this webinar for making it a success. Once again, I thank one and all present in this session. Thank you very much. This is the end of our first technical session. Our next technical session will start at 5:15 p.m. Please join with us. Thank you.
Hello, Sudeep.
হ্যাঁ
Sudeep.
ঠিক আছে হ্যাঁ হ্যাঁ
Just announce before we
thank
take
you so
next
much.
session.
The technical session 1 is over now, and we are now will be again live on this channel at 5:15 p.m. today for technical session 2.
Submission of feedback form for the other
লাইভ বন্ধ করো এক্ষুনি লাইভ বন্ধ করো অভিজিৎ